So I'll just give a little bit of background. It's quite an eclectic conference with an eclectic group of people with background. So I thought I'd give a little bit of background. So basically what I'm talking about is trying to induce oral immunity instead of oral tolerance, and we're looking at doing that in the very young. So the default response to antigens encountered by mouth is oral tolerance. And by definition, then that means re-exposure to that antigen, even later on in life through a systemic route, gives you oral tolerance, this controlled immune response instead of oral immunity. This is entirely sensible. Our bodies are taught to ignore or have a muted immune response to anything taken by mouth. So the environmental antigens, food, commensal flora, all of that is not attacked, and we don't have this rampant inflammatory response all the time. However, it's a catch-22 because most of the pathogens we encounter through a mucosal or an oral route. So that means that if they're not challenged, then basically we don't get immune response until after colonization or penetration. And using military parlance, if you look at systemic vaccines, they're like the soldiers. They're inside a compound, and they don't respond until the enemy breaks down the front door. But a mucosal vaccine will also patrol the perimeter as well as inside the compound. So it would be very valuable to have a good, strong oral immune response to specific antigens. Also, oral immunizations, highly sought. They're needle-free, easy to administer. You don't have to have a specialized person to administer them. So it would be very nice to have. So how do we do it? We're saying in our minds that you must get the antigen across the gut wall to be able to do that. And how do you do that? Well, it's difficult. There's several physical barriers in the body that prevent antigen uptake. There's the acidic pH in the stomach. There's peptidases in the small intestine. There's strong mucus production. All of that is going to stop antigens getting across the gut wall. What you could do is introduce strong mucosal adjuvants such as cholera toxin and its subunits, but that obviously has difficulties. Some colleagues, I imagine, in the next couple talks would probably talk about tethering your antigen to a receptor, which then helps it to get across the gut wall, is very advantageous. You can get a mucosal immune response that way. But for us, we started thinking, well, maybe we can introduce the antigen when the gut is in a semi-permeable state. And I'll explain that in the next slide. And that's for the neonatal piglet and ruminant gut. It's semi-permeable right after birth. So we're saying that if the antigen can get across the gut wall when the gut is leaky, can it promote oral immunity instead of oral tolerance? So piglets and ruminants, sheep, cattle, horses, etc., do not share maternal circulation in utero. They're born immunologically naive. So they must ingest colostrum within 24 hours after birth or they'll die from infectious diseases. The components in colostrum, immune factors such as cytokines, antibodies, hormones, as well as maternal cells, those get passively transferred to the neonate, so you get antibody-mediated as well as cell-mediated immunity. And the gut, it realizes this. So the gut, if you're going to personify it, it realizes that it needs to facilitate the uptake of the colostrum components. So it's designed to be leaky for the first couple days after birth to facilitate that passive transfer. So we started doing our work with vaccinating lambs. So the vaccination regimen here is we gave an oral gavage within the first 24 hours with OVA. We just used ovalbumin at 0.023 grams or 2.3 grams or saline. We've vaccinated again at 28 days of age, and this time we used 10 mg of OVA with incomplete Freud's adjuvant. And then a couple weeks later, we euthanized them, collected blood, lung lavages, as well as the mesenteric lymph node cells. And we see here, if we're looking at the OVA-specific IgG titers in the serum, If we look here at day 0, there's no antibodies against OVA in either group, or actually in this one as well, meaning we're not getting a passive transfer of OVA-specific antigen from the mom. At 4 weeks of age, so they've had the oral gavage, but for a month already. If they were gavaged with 2.3 grams of OVA, they don't develop an immune response. If they were gavaged with fold less, we are seeing induction of anti-OVA IgG in the serum. And at 7 weeks, so like at 4 weeks of age, right, we bleed, and then we do an IP immunization here with the 10 mgs of OVA, and we're getting some immune response here. But again, if they were gavaged with that low dose of OVA, you're seeing a much stronger immune response. And this is the IP control, so saline at birth, immunization at 4 weeks, and then we're getting a stronger immune response. If we look at, this is the anti-OVA IgA titers in the lung lavage. So if they were gavaged with 2.3 grams of OVA, you get a low level of anti-OVA IgA in the lung. But if at 0.023 grams of OVA, again, it's a much stronger IgA response. And the IP control, we got 2 animals showing a response and 2 not, which is interesting. But what we see here is that the cell-mediated immune response is actually very, very weak. So so we've got the mesenteric lymph node cells stimulated ex vivo. So there's just in media or in the presence of OVA, no induction of interferon gamma, and only induction of from one animal with the when you're given the low dose, as well as the IP control. So we're seeing an antibody-mediated response, but not a cell-mediated. So vaccination in early life with OVA alone induced a priming of the oral immune response instead of tolerance when OVA was given at a low dose. So then we move to piglets. They're fantastic for using as a model for the human animal, as well as they're considerate enough to have multiple piglets instead of lambs, which will have 1, maybe 2. So pigs, we made the switch. And here we did the same regimen, except the OVA was gavaged alone or with adjuvants. And this, we've got CPG, which is a TLR9 adjuvant, as well as polyphosphazine, which induces the inflammasome. And then we boosted, instead of using IFA, we use montanide. The reason why we did this is because we can, the animals, if we can get permission to keep them in the food chain, if we used, you can't keep them in the food chain with IFA, with montanide. It's already approved adjuvant for use in pigs. So you're looking at $10,000 worth of difference for a trial. So this is probably a $15,000 trial. But if you can keep the animals in the food chain, it's only a couple thousand. So again, you can't see that down there, so I'll just move on. So if we look at the anti-OVA IgG1 titers in the serum, so again, the titers are much, much lower because it's with IFA, it's a much stronger response. But these animals were given saline at birth and then saline a month later, or IP, which is saline at birth orally, and then OVA plus montanide at 1 month of age. With these guys, they were given 5 mgs of OVA or 0.05 mgs of OVA. And if you were just given OVA alone, they had, in contrast to what we saw with the lambs, they actually have an induction of oral tolerance. If we add CPG and the polyphosphazine adjuvants, we're getting an immune response which is comparable to the IP control group. If we look here with the IgG titers, again, if we have 5 mgs OVA alone, we get a tolerance response. If we have the higher dose, or actually the lower dose as well, in the presence of the adjuvants, we're seeing priming of the oral immune response. And with IgA as well. So oral tolerance with 5 mgs OVA or 0.05 mgs OVA, getting induction of the oral immune response when adjuvants were included. So we're seeing some difference between the animals here. Piglets vaccinated with OVA alone induce tolerance. Inclusion of adjuvants promote priming of the oral immune response. We repeated this again when we had CPG and polyphosphazine, and this time we formulated it as a microparticle. And again, we showed a similar immune response. So we conclude that the oral gavage with OVA plus adjuvants promotes oral immunity in response to antigen exposure in later life. And these are published in BMC Vet Research and then VII as well. So we started kind of thinking about the mechanism here. How is the antigen getting across the gut wall? And I'll just talk about it very briefly. But basically, when you're looking at the villi here, the enterocytes lining the outside of the villi, in the neonate, the fetal enterocytes, they take up antigen by non-selective pinocytosis, and the animals undergo this gut closure, which is the time after which the intestinal epithelial cells no longer take up or internalize macromolecules by pinocytosis. That's the definition. But we can see that it actually it's regional specific. So at 36 hours after birth, and these experiments were done in the late 70s, we see that if you look at just 40% of the small intestine is closed, it's undergone gut closure. After 3 days, the amount of the gut that's closed to pinocytosis is 60%, and to get 100% gut closure, it's actually, they say it takes 18 days. So when people say gut closure, they often talk about 1 or 2, maybe 3 days, but actually it's a regional specific response there. It's kind of neat if they took a little piece of the ileum here and they moved it up to the jejunum, and they found that little piece, I don't have it shown here, but a little piece of the ileum still acted like ileum. It was still open. Even though the environment suggested that, or the environmental that you placed it in was a closed gut. So it's kind of interesting, I thought. But regardless of wherever you're looking in the jejunum or the ileum here, the macromolecules, if they were or were not pinocytosed, after 2 days are not transported to the blood. So, but that's a difference in gut closure. They're not talking about that usually. So what we did was we looked at, in today's animals, I think you'll agree that in 30 years ago, the pigs the genetics are quite different. So we looked here at we did Cy5 labeled ova, and we gavaged that into piglets prior to suckling, and we harvested the organs 6 hours later. And you can clearly see here, I don't know if you can see with the lights, but the Cy5 ova is actually within those enterocytes, so suggesting that that's pinocytosis in the ileum. But if you looked in the first 25% of the small intestine, it's actually, you can see that the antigen is actually in the lamina propria. It's not in the enterocytes. So it's suggesting that it's a regional difference of mechanism of antigen uptake. It's quite distinct. And we do have some facts analysis showing that the enterocytes at the ileum have the ova, but the enterocytes in the jejunum does not. We're extending this time course, but we're quite intrigued by those results. So we're thinking, how can there be, what other mechanisms of antigen uptake could there be if it's not by pinocytosis? So we decided to take a look at the tight junctions. And this is what a tight junction, it's a shame you can't see it quite as well. It looks fantastic on my screen. But if you can see here that right in between the enterocytes, it's actually right on the lateral membrane. It's quite impressive that the claudin, this is antibodies against claudin 4, it's very, very strong in between the cells, which is where the tight junctions will be. But, and that's what we expect to see. But if we look at the 24-hour, this is the jejunum, at 24 hours, actually claudin 4 is right on the extreme apical tip. Thank you. So this is what it should look like, or what we expected it to look like. And actually, the claudin 4 is right on the apical tip of the enterocytes at 24 hours of age. It's not where you'd expect it to be within the tight junctions. At 48 hours, same thing. And then at 3 days, you're starting to see it's actually moved away from that apical tip, and it's in the lateral regions of the gut. And this is what you see at 6 weeks of age. So again, this is the jejunum. If we look at the ileum, at 24 hours, its claudin-4 is within the lateral membranes. It's where you'd expect to see it, and that stays the same over time. So for conclusion, we saw when we gavaged lambs with OVA after birth, we saw induction of oral immune response. In pigs, we saw gavaging them with OVA triggered oral tolerance, but if we included the adjuvants, we saw induction of oral immunity. The piglet and the ruminant gut are leaky for a limited time after birth. And our data, and sorry, I didn't show that, at the 0 hour, when I had the Sci-5 labeled OVA, it got across the gut wall at 0 hour, but at 24 hours, it was nowhere. It was not in the enterocytes, and it was not in the lamina propria. So that could be a difference in how the genetics of the animals have changed, perhaps, over these last 30 years. That the gut is closed to antigen uptake within 24 hours. But we're suggesting that the tight junction formation may not be fully formed after birth, which may facilitate the antigen or colostral macromolecule uptake. So then future work, we're expanding, we're moving past OVA to we're working with Lawsonia, looking at orally gavaging piglets, and then actually seeing if we can see protection. We're doing immunohistochemical analysis with many other of the tight junction proteins. It's a bit challenging finding antibodies that'll work in the pig. The mouse, it got a much bigger toolbox, but we're making some progress. We're expanding our time points to look at weaning, because right immediately at weaning, the gut is in a semi-permeable state as well. And perhaps looking at oosing chambers to look at paracellular transport. And we're also monitoring other movements of antigen across the gut wall as well. And the acknowledgements, I'm at VITO, the Vaccine Infectious Disease Organization, and that's home of Intervac, the International Vaccine Center. So we do we've got level 3 facilities for pigs, horses, cattle, that kind of thing. So it's quite fun, actually. This is my group and previous members, and funding from Ontario Pork, Alberta Livestock and Meat Agency, and Agriculture Development Fund from Saskatchewan. Thank you.
Can you comment on the dosing effect? Because I saw that in the lamb study, used this low dose inducing response, but the high dose did not. But in your pig study, it seems like both low and high dose with adjuvant induced immune response. Can you give
if we had tons of money, we'd do a real nice dose kinetics, but you just almost have to kind of pick a dose and then try to work above or below. And we did some initial work with rat pups, and we extrapolated the result, the body weight to the lambs, and then and that's why we have those funny numbers like 2.3 grams of OVA. And then we tried to do a similar thing with the pigs, but we saw that the really, really low dose seemed to work better in the lambs, and both doses appeared to work fairly well with the pigs. Others show that too. I mean, if you have a really high dose of OVA in other animals, you're going to get oral tolerance through one mechanism. If you have an extremely low dose, you get oral tolerance through a completely different mechanism, and the sweet spot is in between, and you can get oral immunity. So we're just trying to find that dose. But as to explaining the mechanism, I'm sorry, I don't know.
Yes, because there very interesting study actually showed that when you prime the animal with ultra low dose antigen, you induce a very high pro-inflammatory response. If you give an intermediate dose, you can activate both inflammatory response and regulatory response. Mm-hmm. So you'll see a nice
immune response. But if you give a dose very high, then you induce regulatory response.
Exactly.
And so
it's quite
there is a
interesting.
very interesting mechanism into it.
Yeah.
Okay, one more.
I don't remember when you exactly did the intramuscular boost. Yes. When did you do the intramuscular boost?
Actually, with the lambs, it was an IP, and that was at
okay.
28 days of age.
And in the pig?
And the pigs, same. It was 28 days, but that was
Yeah.
IM. That's right.
Did you do the same? Did you do the same in older pigs? Because I'm quite sure if you give these amounts, it's the same as the previous question. If you give these amounts in younger pigs, you also get priming. In older pigs, you get priming of immunity.
We're doing those experiments right now. We're making this assumption that it is a gut closure event, but we haven't actually done those experiments to prove that. So that's actually the pigs are farrowing today, I think. So.
And you speak always of oral immunity, but you show serum antibodies.
Yeah, and so I looked at the lung lavage to look at the mucosal immune response. So an oral vaccine, right, will get a response in the blood as well as to the other mucosal sites.
Not correct. You can have passage of your antigen and reach the systemic immune system and not having a local immunity in the gut. So you have to check there also.
Yeah, we'd like
Okay.
to, yeah.